Hi, my name is Jason Grant. This is my gut microbiome presentation. So first off, a gut microbiome consists of the types and proportions of bacteria found in a location, specific location. So the gut microbiome specifically is the diversity and distribution of bacteria within the gut of an organism or animal. Some physiological conditions related to the gut microbiome. It's related to a couple different digestive disorders, some psychological disorders, specifically. It's been linked to Crohn's disease, recurrent CDI infection, irritable bowel syndrome. In patients with the CDI, performing a fecal matter transplant on the patients greatly diminished the likelihood of another outbreak. This is because they replaced the bad bacteria with good bacteria, and then the overall health of the gut increased. Some psychological disorders that have been linked to the gut are depression. Studies show correlation between the organisms and microbiota found in the digestive tract. So all the different bacteria found in the digestive tract actually have an effect on those with depression. Those with depression had a different species of bacteria found in their stomach than those without depression. So our experimental question was, will there be a significant difference in the gut microbiomes between two different species of animals? The animals we picked would be two guinea pigs, which should be similar, and then the contrast we picked was the iguana. We think that the microbiomes of the two guinea pigs will be similar, and they'll be vastly different from that of the blue iguana. So here's a little chart displaying the characteristics of the two animals. So we have two guinea pigs, same age, same weight, both males, both in very good health, have the same diet, same supplements, same exercise level. And then we have the blue iguana, which is only 6 months old, only weighs 2 pounds, also male, very good health, completely different diet, no supplements, and only occasional exercise. So there's several steps involved with this. The first step would be to purify the DNA from the fecal matter, which we use a Quiogen kit for. It's specifically designed for the isolation of DNA from fecal matter. We followed manufacturer instructions, and we concentrated the DNA following the manufacturer's instructions. We use a Zymo kit for this and utilize a spin column filter to catch the DNA. Then we quantify the DNA. We use the nanodrop spectrophotometer. It measures ultraviolet absorption, in this case specifically at 260 nanometers and 280 nanometers. This gives you a ratio of the absorbance at 280 and 260, and it tells you how pure your DNA is. Pure DNA should have a ratio of about 1.7, but anywhere from 1.5 to 2.2 is considered acceptable. If it's any higher than this or lower than this, it's either contaminated with proteins or RNA, other things that aren't DNA, which is what we were looking for. After we did this, we amplified the DNA. We used the amplicon PCR reaction, so a polymerase chain reaction. It amplifies the 16S ribosomal RNA gene, specifically V3 and V4 variable regions, because those regions differ between different species of bacteria, while the rest of the DNA sequence stays about the same. So you can identify your bacteria present from this region of DNA. So after that, so the cycle for this, we did 30 to 40 cycles of replication. Step 1 was 1 minute at 94 degrees Celsius. Step 2 was 45 seconds at 54 degrees Celsius. And then step 3 was about 2 minutes at 72 degrees Celsius. Then we moved on to the gel electrophoresis. This made sure that our PCR was successful. 250 volts were applied to the DNA samples. DNA is positively charged, so it moves from the negative side towards positive. And then the distance traveled is actually based on the size of the molecule. After we ran the gel electrophoresis, we did a PCR cleanup. We used the Ampure XP kit by Agincourt. It removes all the buffers used in the amplicon PCR reaction, which would be like magnesium, some of the primers, the enzymes, just anything left over that's not DNA at this point. It also removes the unused primers, like I said. Removal of these molecules was vital to the DNA indexing step, which would occur in the next step. Any leftover buffers or primers can interfere with the indexing. So next we indexed the PCR, and that was used to tag the specific DNA samples from each group, so sequencing could be done in one batch. So the thermal profile for that was 95 degrees Celsius at 3 minutes, 8 cycles of 95 for 30 seconds, 55 for 30 seconds, and 72 for 30 seconds. And then last was 72 Celsius for 5 minutes, and then you hold it at 4 degrees Celsius to keep the DNA from degrading. Well, and after you do the indexing, we did one additional PCR cleanup to remove anything else that could be left over besides for DNA. The same technique is stated in this cleanup. So then we wanted to quantify our DNA PCR indexing PCR product. So we use the fluorimeter, and the qubit fluorimeter actually, and it measures the concentration of amplified sample, making it possible to determine the correct dilutions for the sequencing reaction. It's more accurate than the spectrophotometer previously used, and it can detect smaller amounts of DNA. And then we sequenced the nucleotide. It was the nucleotide sequence was determined using the Illumina MISQ or MISSEC next generation DNA analyzer. So this table here is the results from the spectrophotometer. You can see the DNA concentration on this line here. 408 was 31.7 nanograms per microliter. We had 60.3 nanograms, 39 nanograms, and then 27.7 nanograms. And as you can see here, all of our 260 to 280 ratios were within the acceptable range, meaning that we had relatively pure DNA. So we have the fluorimetry results here. We'll talk about gel electrophoresis next. So the gel electrophoresis to make sure the PCR reaction was successful. We have a mark for a positive control as expected. There's no mark for negative control, which we wouldn't expect to see because there's not going to be any DNA or negative control. 885 actually did not have a mark, which means that the PCR was not successful. But our other samples, they did have a mark where expected, which lined up with our positive control. And then this bar here would be the standards used. Back to the fluorimetry. So our positive control, we actually did not have enough concentration for the fluorimeter to pick it up, which was unexpected. And then our negative control, you would expect to not see any concentration because there shouldn't be any DNA in it. And then 885, there was also nothing that was shown by the fluorimeter, but that was expected because, as you can see here, we didn't have any results from the PCR reaction. Everything else, we actually had enough of a concentration of DNA to do our dilution so we could be used in the sequencer. So as you can see here, we had to dilute our sample with the highest concentration to 30.9 nanomolar. 408 was 3.3 nanomolar, and our second 408 was 17.6 nanomolar. So this graph here is the kingdoms that we found. We found it was about 99.5% for all groups for the bacteria kingdom. There's only a small amount of other. Other would just be any other materials that were in the DNA that weren't bacteria DNA, anything unidentifiable. Then we have the order classification. It's kind of hard to see from here, but there were only a few classifications where all three animals shared similar ones. But for here, you can see there was 2 classifications that only the guinea pigs had, and then these 4 here are 4 classifications of bacteria that only the iguana had. So there were a couple classifications that were shared in the order, but when you break it down to the genus, they actually had no similar genuses. The only similar ones were unclassified or other, which couldn't be detected by the sequencer they were using or the analysis. But there is actually 4 kinds of bacteria that were only found in guinea pigs, and about 6 kinds of bacteria that were only found in the iguana. The guinea pig and the iguana did share one strain or one genus of bacteria. So we're going to move on to the discussion. Based on spectrophotometry, DNA was successfully extracted from all experimental groups. We had 2 replicates for each of our experimental groups. So not all replicates were successful, but at least one replicate of each group was successful. The 260 to 280 ratios for all samples was also acceptable. The PCR was not successful for 885X since it didn't have any sign on the DNA moving up the plate, the gel electrophoresis plate, which was right here. It is expected to not see mark at negative control as stated because there shouldn't be any DNA in the negative control. The results of the indexing PCR and PCR cleanup showed that there was an issue with the positive. There's actually no issue with the positive control. There was only an issue with 885. So the other samples, they actually had a high enough concentration to be sequenced. You can see that in the gel electrophoresis, and then once again in the fluorimetry results. Our negative control didn't show any concentration of DNA, but if it did, you would expect cross-contamination. So the positive control, or for our DNA sequencing results, this is actually the number of reads right here. The guinea pig had a high number of reads. So all of the experimental groups had a high number of reads, as well as the positive control, which is expected. And the negative control had a low number of reads, which was also expected. Negative control should have a low number of reads because the negative control should not have any bacteria in it. And the reads correspond with bacterial DNA that has been picked up by the analyzer. So our positive control, it's sold as E. coli bacteria. However, no E. coli was found in the positive control. The positive control is actually consisted mostly of Escherichia alberti and a few other trace bacteria. This indicates cross-contamination and possibly mislabeling by the supplier. So the number of reads for the animals being compared was more than sufficient. All three animals had the presence of the Bacteriadales and Clostridales order. Both guinea pigs contained Sphingobacteriales and Spirochaetales, while the iguana did not have these present. The iguana had 4 different kinds of bacteria present that the guinea pigs did not. And then on the genus level, this actually broke down even further, and they had more dissimilar. The 2 guinea pigs, gut microbiomes are more dissimilar than the iguanas on the genus level. One bacteria that was found in the guinea pigs would not be iguana. One genus would be Treponema, and this bacteria is mainly responsible for syphilis. So our hypothesis, it was supported by our results, and as seen in the graphs, the 2 guinea pigs had extremely similar gut microbiomes, while the iguanas had pretty much completely different genuses of bacteria present. And that's it.